Hello, everyone. Good evening, good afternoon, good morning from wherever you're listening in today. I'm really pleased to be able to present to you a webinar with Sarepta Therapeutics, and we're going to talk about the recent events and that's occurred in Sarepta Therapeutics. And we're joined by Wendy Erler, Doug Ingram, James Richardson, and Dr. Craig McDonald. I think that the decision to participate in a clinical trial or to take advantage of a therapy that has been approved is always a difficult one. We as parents and individuals with this diagnosis have to think through both the benefits, the risks, and that matched against a natural history of this disease that is well known and that we carry in our hearts from the day of diagnosis. None of this is easy. And I've heard from so many parents who say they want to make the right decision. And in therapy, because of the variability, because we're human beings, because of the diagnosis and all of the genetics, the right decision is a hard one. And I think that we spend time and hope and some joy thinking about this 24 hours a day, 7 days a week, before the trial, during the trial, after the trial, and certainly with regard to an approved therapy. So with that, I want to keep that in mind, that we're all warriors here. We're all people who've given blood, sweat, and tears to make progress in this community. And we are a robust, vibrant community that cares deeply about our children, those individuals who are diagnosed with Duchenne. We want this to be better. We want therapies to extend not only function, but the lifespan of those we love. So with that, we're going to talk about AAV gene therapy, which, as you know, is probably here to stay in our world for a period of time, maybe for a very long time. But this has been a very difficult week for all of us. And so with that, I'm going to introduce Wendy Erler. We will go through slides. We will come back with those slides with questions once we talk about the slides. And once Craig McDonald and I have a little discussion, then we'll talk about your questions. Keep in mind that the PPMD conference is this week as well. Sarepta will be on site and will be very willing to entertain questions and about this therapy and their plans for next steps. So with that, I'm going to turn it over to Wendy Erler. Welcome, Wendy.
Thank you so much, Pat. Hello, and thank you everyone for joining. As Pat said, my name is Wendy. I lead patient affairs at Sarepta. I'm joined today by my colleagues, Doug, who's Sarepta's Chief Executive Officer, and our Chief Medical Officer, James Richardson. We really do want to use our time today to share with all of you in real time what we are learning specifically about ELEVIDYS in light of a recent safety signal. And we also want to just remind everybody that this is truly real time. This very tragic event just happened. So there will be unanswered questions. There will be things that we just don't have answers yet, but we are all working very hard to provide transparent and clear information. Most importantly, we want you to hear from us from the bottom of our hearts that we share the deep grief of the community at the loss of a second patient who was treated with ELEVIDYS. One patient is one too many, and we really, really do share in the trauma of this news, and specifically for the family, we are, and I say this sincerely, everyone's in our thoughts and prayers. Today, the purpose is really to share what we know, when we knew it, and what we have learned. What informs our recommendations as the company? So, there are pieces of this that are informed by regulators and by physician experts. So, everyone has different pieces of the work to do. But what's informing the recommendations from Sarepta? And then understand your questions. If we can't answer them all now or later on this week in the conference, what information do we need to continue to get and provide? Our commitment has always been and will continue to be transparency and being very clear in what is known, particularly what's been driven by the data. We shared in our last webinar all of the publications and efforts that have gone into making data public. That continues in light of all pieces of news. I'm really delighted to turn this over to Doug, who is our CEO. I've seen how Doug has personally reacted to all of this news, as well as from a company perspective. And in the Duchenne community, we're lucky to have Doug at the helm. Doug?
Well, thank you, Wendy. I'm not sure I deserve that, but thank you very much. And Pat, certainly thank you for hosting this, and for everybody that's participating, thank you for spending some time with us as we talk through these issues. I think I'm going to try to be brief in a moment. I'm going to turn this webinar over to our Chief Medical Officer, Dr. James Richardson, and he'll discuss things in more scientific detail than I certainly can. And then there'll be a discussion with DMD specialist, Dr. Craig McDonald, and we're really honored to have Dr. McDonald on this call today as well. But before we do all of that, what I would like to do is review the recent news, make sure we're all on the same page about the steps that we're taking to ensure that we prioritize patient safety and perhaps give a little bit of context. As I'm sure all of you know, yesterday we announced that last week, really only a few days ago, a non-ambulatory patient passed away from acute liver failure following treatment with ELEVIDYS. This was a tragic event for this family who, with ELEVIDYS for the first time, had objective reason to believe in a brighter future for their son living with this horrible, degenerative, and invariably fatal disease. And this followed a similar outcome about 3 months ago. It was a tragedy, of course, for the family, and it is heartbreaking for all of us who have dedicated these last many years to an effort to improve the lives of DMD patients and the families of DMD patients. Now, how we should react in response to these events was not a simple calculus. And let me try to provide you with a little background to explain why. A couple of things to know. All muscle-directed and full-body infused gene therapies today are delivered using a capsid. It's called AAV. I'm sure Dr. Richardson will discuss more about this. AAV can place stress on the liver and comes with a rare risk of liver injury, becoming acute liver failure in the first months after gene therapy infusion. That's a reality. However, ELEVIDYS has been regularly infused for the last 7.5 years. I think its first infusion was in January of 2018, and there was never until these last two events an incident such as this. About 950 patients have received ELEVIDYS over the last 7.5 years across a broad ranges of ages and weights and ambulatory status. About 150 of those were non-ambulatory, about 800 of those were ambulatory. Now, over 30% of patients see elevated liver enzymes with AAV generally and with ELEVIDYS in particular, and that's a marker of liver stress. But over those 7.5 years, they were always treated conservatively with a modest increase in steroids, and they came back down to baseline in the first couple of months with no consequence. It was only with these two patients that a signal of liver failure arose, and even with these two recent events, the risk of liver failure remains very rare, and for now, the signal is no higher than other approved full-body infused gene therapy. So with that background, one could arguably have concluded that as the risk-benefit remains positive for ELEVIDYS, given the signal, no changes should be necessary. However, as you know, we did not conclude that. We concluded that we need to make some very significant changes, and we need to make them rapidly. You see, Sarepta had completed a preclinical non-human primate study using a form of immunosuppression treatment called sirolimus. And that was used as a pretreatment for AAV-mediated gene therapy. And as a result of that, that indicated, at least in a preclinical model, that this has the potential to significantly reduce the risk of elevated liver enzymes and therefore liver failure in patients treated with AAV gene therapy if, in patients, it operates the same way that it is operated in non-human primates and the way it is operated in other gene therapies, at least in individual case studies and some anecdote. Now, let's talk about the calculus. ELEVIDYS works by delivering a gene that codes for a form of dystrophin intended to protect existing muscle. The goal then is to arrest decline and further damage, but what it cannot do is bring back muscle already lost to Duchenne. So the safety team, when evaluating this, had to carefully consider the very real harm that will come to non-ambulatory patients from a delay in receiving ELEVIDYS as soon as possible. But against that, they had to consider the opportunity to reduce this risk of liver failure in non-ambulatory patients. And they quickly determined, very quickly, that a temporary delay, painful though that is, for patients who don't have the luxury of time, and many patients who were scheduled for therapy and had built their lives around getting this infusion, they concluded that harm was worth it to the broader community and to these patients to put in place a new protocol that would use sirolimus in connection with future treatments for ELEVIDYS. As a result of that, we've done the following. I'm sure you know this generally, but I'll go through it again, and I'm sure James will talk about it in more detail. With respect to commercial ELEVIDYS, we have decided to pause shipments for non-ambulatory infusions until we have completed two things. One, we've held an expert panel. So we've taken advice and guidance on the exact pre-treatment protocol to employ. That's a very, very important step. And second, we need to speak to the FDA. We need to take the FDA's input and get concurrence with the FDA on that protocol. And if we can get all of that done, then we would like to start making infusions available commercially. The second thing we did was, with respect to our non-ambulatory trial, ENVISION, we have paused dosing to complete a protocol amendment to add that sirolimus pre-treatment protocol there. That is a very painful thing to do. You know, having been doing this for a long time, one of the real painful histories of Duchenne muscular dystrophy is that the non-ambulatory patient population is often left behind historically. And so, you know, I've been very proud of the ENVISION and the opportunity to do clinical trials with non-ambulatory patients, but we're doing the right thing, and the FDA concurs with it. I cannot tell you, as I sit here today, precisely how long those things will occur, but I can promise you that we will move as fast as possible to get all of this done and in place, and we will keep the interest of patients firmly in mind at all times. So finally, what I'd like to do is just really talk about the two big priorities here for Sarepta. The first thing that you need to know about us is that we are driven by the goal of helping DMD patients live a better life. I myself have been single-mindedly committed to a better life for Duchenne muscular dystrophy patients for the last 8 years. And the commitment that I have is shared by all of my colleagues at Sarepta. I don't stand alone in that commitment. This is not merely a business to us. It is a mission. I will mention now, maybe apropos of nothing, something to you this evening that I have never mentioned publicly before. I have often said that I do not have a son with DMD, and that's true, I don't. And so I have always been careful never to presume that I can understand fully your journey, because I haven't lived your journey, and I understand that. But I do need to tell you that I have someone in my very immediate family. There is no human being any more precious to me than this family member that does have a degenerative muscular dystrophy. He has classic myotonic dystrophy type 1. I wish we had an ELEVIDYS for him and others with DM1. We don't, but we're working on a treatment. I hope that treatment will offer the hope that ELEVIDYS offers to the DMD community. I say this all to you, whether you choose to believe it or not, to tell you that this mission could hardly be more important to me than it is. And I should also tell you that the passion that I have for this mission, and it's an unreasonable passion at times, is a passion shared by all of us at Sarepta. I am not alone in this view. And the second thing I want you to know is that we are committed to being authentic and transparent with all of our communications with the community. In fact, compared to most biotechs, we likely over-communicate, and we often do so as early as possible, even when it's not in our best interest sometimes. It's even in our detriment. But if one is serious about being mission-driven, then honesty, openness, and transparency is required, and we are committed to that. With that said, I'm going to turn this call over to Dr. Richardson, who can provide some scientific insight for you. James, I don't think I hope I may not be alone. I can't hear you, James.
I don't see James online. Did we lose him?
No, he's here, and he's not muted, so give him just a second. Doug, while James is getting online, do you want to just speak to the Embark evidence to date around the first 90 days post-infusion while James is getting audio?
Everyone can
Oh,
hear me
yeah.
now.
Oh, good. We can hear now. Go
James,
ahead.
James will be a much better person to discuss that with more scientific authority than I could ever have. So I will shut up and put myself on mute.
Hi, everybody, and thank you, Doug. I hope you can hear me okay. I've had to switch to another microphone. I'll try and speak up. I just wanted to start off by echoing Doug's final words there, that this really is a mission for us at Sarepta to support those living with Duchenne. It's what gets me up in the morning and what stops me sleeping sometimes at night. And I think top of that list at the moment is safety, and making sure that that always remains top of our list and an absolute top priority, and something very specific and important to what I do at Sarepta specifically as well. So I'm going to talk a little bit about acute liver injury and liver failure, and I think the distinguishing between those two can be helpful a little bit to understand some of the data that both we talk about and perhaps what's also on things like the FAERS database. Acute liver injury, as Doug has said, is a well-known phenomenon in AAV-mediated gene therapies. We see it and have always seen it in ELEVIDYS at relatively rates of between sort of 30 and 40%. The majority of these cases, these patients have been managed through that period of elevated liver enzymes as an outpatient. But there is, and it always has been consistently, a subset of more severe liver injury, as kind of indicated by the level of the blood test changes that have had to be admitted to hospital normally to receive intravenous steroid treatment. Prior to March this year, all of those patients, whether outpatients or inpatients, have recovered from their liver injury. So I think in addition, there's a number of questions about timing of liver injury, and we'll talk a little bit about liver failure and timing as well. And this slide is helpful as it's data directly from the Embark study. That was the randomized controlled study in 4 to 7-year-old boys. This is data over 2 years, because the study, although read out at 1 year primary outcome, we managed to follow it out for a 2nd year and collected adverse events, side effects, let's say, in that period as well. The rows on this table are the terms that the investigators used to describe these adverse events. So we're going to talk about acute liver injury as a term throughout. In terms of this table, I just ask you to focus on the line that says GLDH increased. That's a very sensitive blood test normally used just in clinical trials to detect liver injury. And then gamma glutamyl transferase, often better sort of known as gamma GT increase, and that's a blood test that you'll more commonly hear about in clinical practice. So those are the two important lines for this table. And then the columns are when these events were first, when these events were ongoing within the study. And the first column marks an initial 2-week period after dosing, and then as you move to the right, you get further out until you end up at 2 years. So you can see that the majority of events occur in that first 2 weeks post-dosing, and that's specifically the GI side effects, the vomiting, nausea, decreased appetite. This list just has the most common side effects. This early period is the time we've also seen troponin rises. We don't have them here, but in other studies and in the clinic, this is a very common period for myocarditis and troponin rise. The liver changes you can see are almost all in that 2 to 60-day week period, 2 weeks to 60-day period. So it's very, very consistent that we see the onset of acute liver injury in sort of 4 to 8 weeks after dosing. And it normally also resolves in that period. And if you follow the table out to the right, you see very few events after that 60-day period and particularly after that 12-week period. And although we have one GLDH increase ongoing at 12 to 52 weeks, we don't have any patient within our clinical trials that's developed new liver injury outside of that first 90 days. We'll move to the next slide.
James, before we move on, I'd like to take the opportunity to assure everyone that's listening tonight, because there's been some comments circulating online via what is social media, there is absolutely no world in which Sarepta is hiding data or not sharing data. These data have been presented and published and are very, very much attributed to the scientific process that is clinical research. There are very distinct safety reporting requirements, and Sarepta adheres to all of those. So we wanted to review this data just to add confidence that this is really what we know today, particularly from EMBARK.
Thank you, Wendy. Just to talk now a little bit about acute liver injury versus acute liver failure. So acute liver injury, we define and we detect by measuring liver enzymes such as these transaminases, ALT and AST are the two most common, what we call transaminases, which are enzymes that go up in liver injury and really indicate liver inflammation. That's distinguished from liver failure, which is a more severe phenomenon in which the liver actually loses the ability to produce important proteins. Most important proteins that we see are things that help the blood clot, and a protein called albumin, which helps keep fluid from leaking outside of the blood vessel. It keeps the fluid inside the blood vessels. Liver failure, as opposed to liver injury, is defined by tests looking at these proteins that show low levels, and then also detect that fluid that's leaked out because of the low levels of albumin in areas where you wouldn't normally expect fluid. So one of those is the abdomen. And on this slide, that's a medical term, it's ascites, but that's essentially fluid in the abdomen that's leaked out because the albumin is low, because the liver is not making albumin at a sufficient level. Many times, liver can recover from this failure state and start to make those proteins again. Sadly, that didn't happen with these two patients who passed away. I wanted to just sort of tackle the question around ambulatory status. I think it would be just very clear to say we have not seen cases of acute liver failure in ambulatory patients, but we do see acute liver injury. And it's not possible to exclude the possibility that we may one day see an acute liver failure in this ambulatory population. And I think it's important to consider this potential risk when people are deciding on a course of treatment. We took very specific urgent action because we saw a definite signal, as we call it, these two patients who were non-ambulatory, who passed away of acute liver failure. But we remain attentive for developing signals across the whole population. It's extremely likely, well, not just likely, it is certain that the ability to walk is not what is driving what we see as an increased risk of liver failure. Rather, that's a surrogate, a marker for something else, some kind of underlying predisposition that comes with disease progression and age. We want to do a better job of defining that for you, and we are working with urgency and diligence just to try to identify a population more clearly who are at increased risk, whilst continuing to monitor the whole population for any new safety signals. What we are aware of, I think, is that there is a development from acute liver injury, these two patients that we've seen, into acute liver failure. And we don't know what it was that caused these two non-ambulant patients to develop acute liver failure that we haven't seen elsewhere in these 900 patients that we've dosed. But we do believe that if we can modify the chances of developing the initial acute liver injury, we should be able to modify the risk of progressing to acute liver failure. And to do that, we have to tackle what we believe is causing the underlying problem with acute liver injury. Our understanding, both more broadly from the field and what we've observed, is that the initial liver injury is being caused by an immune reaction to the AAV vector, the viral shell that's carrying the transgene that Doug referenced earlier. And it's likely that it's something called a T cell immune response. This response causes inflammation, which we can see in more severe cases manifests as that acute liver injury, and then which we believe in these two cases progress to acute liver failure. This is an immune response driven by something called the adaptive immune response. Next slide. The adaptive immune system is something that we all have that learns to recognize new pathogens or enemies and creates an immune response directed at that specific enemy. The adaptive immune system, in contrast to the innate immune system reacts very quickly. Things like you may have heard of complement and complement-related problems with gene therapy. That's an innate immune response and something that you see very soon after infusion. We see with these liver injuries, these come on that 4 to 8 week period after infusion. And I think that's a very strong clue that this is an adaptive immune response where the body is seeing the vector, the viral vector in this case, as an enemy and causing this immune reaction. We are in the process of convening an expert panel to provide advice on how we can use supplemental immune suppression, as Doug has mentioned at the start, that inhibits T cells and perhaps also these B cells that you saw on the previous slide, which form the core of this adaptive immune response. That hypothesis is driven and supported by the preclinical work that Doug referenced. We looked at a potent T cell inhibitor and a drug that can also help minimize the formation of antibody formation. And that was an experiment, as Doug described, done with non-human primates, looking at the use of an experimental version of a drug called sirolimus, and the ability of that drug to reduce the formation of antibodies and to reduce the acute liver injury. I think there's a question just before we sort of finish up around, well, if we had this work, why are we talking about it now? I think we have to be clear that we have very recently come across this signal of acute liver failure. We did not see acute liver failure in either these preclinical studies or in any of our clinical studies, and nor in the treatment population until this March. It's important to recognize that immunosuppressants themselves have side effects and risks, and these need to be weighed appropriately against their benefits when developing treatment protocols. So that's something that really important piece of information to take in, along with the preclinical studies and other insights from medical practice, into the planned expert committee, who are going to help us develop what we think will be a robust plan to mitigate this risk of acute liver failure. And we'll take that to the FDA and come back to you urgently with the outcome of this expert advice and this regulatory advice. I think, as Doug has said, as I said at the start, this is our mission, and our commitment to patient safety remains unwavering. We are continuing to review these cases, review our data, doing everything we can to better understand this risk. And as we do better understand this risk, we will continue to keep you updated with expediency. With that, I will turn it back to Wendy, and I think we have some time to take some questions coming up.
Thanks so much, James, and thank you for going through the data. And I think it's a good opportunity to thank everybody that did participate in the clinical trials. We know how intense and what just incredible families have done to bring us to where we are today. So thank you to all of the families that have been in the trials. One of the reasons we wanted to focus on when we have seen these adverse events is typically in the first 90 days, is we understand a number of physicians are getting calls from families that have had experience with ELEVIDYS over a year ago, and do they have anything they need to worry about? While we don't know the answer to that question specifically, we do know that most everybody who has experienced adverse events, it's in that first 90 days. So for the concerns and fears that people are feeling now after this last very tragic event, we just wanted to clarify that. And Pat, I'll turn it over to you and Dr. McDonald.
Thank you, Wendy. Dr. McDonald, always happy to have you here with us to answer questions. I think that you're one of the clinicians in the United States who's had the most experience across the board in clinical trials, and certainly with your patient population out there at UC Davis, you have seen and treated considerable number of patients with Duchenne muscular dystrophy. So, Craig, in light of the fact that there are 2 deaths now, and I must say that clearly a lot of people have been treated, but when a death occurs, it breaks our heart, but it also terrifies all of us. So can you talk to me about what are the questions? Because if I'm a new diagnosis or a person interested in this for someone I care about, or for that matter, an individual with Duchenne, what are the questions I need to be asking? What are the things I need to be thinking about now in light of the discussion that we've just had?
No, again, Pat, thanks for the opportunity to participate. And I think with regard to my experience with clinical trials, and I've also had about 35 years of experience taking care of Duchenne patients over the years. And I think, first and foremost, I think it has been, really quite rewarding to be able to, offer as much hope to our patients as we're able to offer them now with this changing treatment landscape. And I think as we approach any of our patients, I think for all of our patients, time is muscle. And oftentimes that lost muscle is irreversible. You don't get it back, and you lose functions that are incredibly important to quality of life. So I think for any of our patients throughout the course of the disease, we have to be really cognizant of what the treatment options are in terms of the treatment landscape, in terms of standard of care treatment, getting good multidisciplinary care. But also, I think it's been exciting that we've had, multiple new drugs approved. Some of them are gene-targeted drugs. Some of them actually address downstream effects of the disease. And we now have many more options to offer both our patients who are ambulatory and non-ambulatory. So I think first and foremost, I think with any of our patients, I think your physicians, will be discussing with you what really the multiple treatment options are in addition to AAV gene therapy. And I think those discussions have to be carried out in a very careful, thoughtful manner, and discussing really what data is out there. I think certainly, we do have much more data available to us in the younger ambulatory population, and we've been certainly gaining experience and learning much more about the management of the older patients with Duchenne, the non-ambulatory patients. I think what's important as I talk to patients about the clinical data is available is what's been the experience from the clinical trials, and what types of patients have been treated previously in the clinical trials. So I think one has to just be cognizant of what the functional status is of the patients. Is there an expectation that we could actually have a meaningful effect in terms of slowing of the progression of the disease? Most of the patients that have been treated in the ELEVIDYS clinical trials have had at least some upper limb function to preserve, some pulmonary function to preserve, and have had cardiac function that has been at least they've had sufficient cardiac reserve, with most of them having ejection fractions greater than 48% and higher. That's not a hard and fast rule, but certainly that's the data that we have available to us to date. So I think it's really important for us to just be aware of what the experience has been in patients that are similar to your son or yourself, for a young man with Duchenne who's considering these treatment options.
Thanks, Craig. Craig, so when we talk about a non-ambulatory patient, can you, just from your perspective, give me the definition of non-ambulatory?
Yeah, so again, I think when somebody isn't really able to functionally stand and take steps across the room, that's generally somebody that for their functional mobility, they're using a wheelchair for all or most of their functional mobility. Some people will define non-ambulatory or loss of ambulation as being sort of unable to walk 10 meters or 30 feet across the room. But I think for the most part, inability to take independent steps across the room would be somebody that would be considered non-ambulatory.
Thank you. I'm going to go back to the slides, and we're going to go back through the questions, and James and Craig and Doug, please feel free to answer them. So I'm going to start with slide 9.
That's what you think, Pat. Hold on here. I can't see any slide numbers. Here we go.
One more. Well, this one will do. But so, you've presented data on the EMBARK study. And I think that when we're looking at these 2 deaths of these young men, this is in the ENVISION study. Do you have data on the ENVISION study that we might be able to better understand? And in tandem with that question, when you see these elevations in the liver values that you've talked about, at what point do you intervene? So you've said they increase, and in many patients across the board in gene therapies, we see liver values increase. So at what point is there a certain level of increase that you begin to think now's the time, and you start either increased IV meds or other medicines to bring that down? Who wants to take that question? James? Craig?
Yeah, I mean, I'm very happy to take the question about ENVISION, and then Craig or I can talk about treatment, and maybe Craig can flip from his experience there. So ENVISION is an ongoing blinded study, and I 100% appreciate I have it myself, the desire to get data sooner from this study. We are obviously challenged by that blinding and the fact that that study is serving as the demonstration of efficacy in the non-ambulant population as asked for by the FDA. What we have been able to do is look at the blinded safety data and compare the frequency of events within the ENVISION study, accepting that half of whatever we're looking at are on placebo, and then go further than that and look at whether we split that blinded data up by age or by weight. And what we're seeing in ENVISION is very comparable rates of acute liver injury between what we expect to see, accepting that this is likely
I think, James, you've frozen for a minute.
I could certainly jump in, Pat.
in
if,
terms of
No,
the
please do,
ambulatory
Craig.
population.
Go ahead, James.
I don't know if you can hear me
Yeah,
now.
I think you broke up there. If you could just repeat that.
Yeah, I mean, I think overall, if we look at the blinded data with Envision, we're not seeing a signal that these events of acute liver injury are happening more frequently in Envision than we would expect it to happen based on what we have seen in our other studies of younger patients. And we're not seeing within Envision there being a difference between perhaps patient weight, for example. And I think that's a really important finding to more broadly contextualize what we're seeing now, is that we're not seeing a differential rate, we don't believe, of acute liver injury between the different ages of Duchenne or ambulatory status. But what we have seen is acute liver failure only so far in this non-ambulatory population. And that's what has driven the kind of urgent action we've taken to preserve, what we, from a safety perspective. But we are continuing to, be open-minded and incredibly thorough in trying to better understand what it is that sort of drove these two patients from acute liver injury into acute liver failure, and how that may be related to their underlying disease state.
So a couple questions about the two patients for both of you in the Envision study. And so in the Envision study, you received an approval last June. I remember it was at our conference last June that the approval, the traditional approval 4 to 6, and then the accelerated approval in the non-ambulatory population. And at that time, you had very limited data, I'm suspecting, although I don't have access to it, but on the few patients that had been treated. And you looked at the data that you had in terms of recognizing, as you mentioned, it was placebo. Now, and I recognize that it is a placebo-controlled study, and therefore the data is blinded. But at this moment in time, given the fact that we've lost 2 boys, are you uncovering or taking a look at that data to see in your Envision study, are you seeing these liver enzymes elevate? How quickly are they elevating? How high do they stay? For what period of time do they stay? And are you really looking at that so you can bring this data to that population that has been approved and seeking access to the ELEVIDYS?
Perhaps we're absolutely looking at the Envision data as it, in a blinded fashion, as it in real time. I mean, we have medical monitors, as any clinical trial would do, who are constantly looking to see if there are patterns within the study that are unusual, or pulling out certain new signals that we haven't seen elsewhere, or anything we may need to do differently within the trial. So we do that as the sponsor. We also share all of that data with the independent data monitoring committee, who are reviewing that data both quarterly, but also if there's an event like this event, this fatal event last week, we'll meet ad hoc to also review the data to try to understand, is there anything that they are seeing within that makes us think there is something worrying or different, and that we need to amend the trial, stop the trial, or unblind the trial, as you say, Pat.
Right.
That's an extreme option, to totally unblind the trial. That will likely render it not viable to support efficacy claims later on, to really, answer that really important question as well, is that this drug is showing efficacy within this population, as well as understanding the safety.
No, I completely understand. So
real quick, Pat, if we could go to slide 10,
Yep.
and I could just briefly address your previous question. But as many know, the AST and the ALT are liver enzymes that fluctuate and are spilled by muscle, and are quite variable in Duchenne patients. And I think the clinicians really focus predominantly on the GGT level, because that's usually normal in a Duchenne patient, and it's really an excellent marker of acute liver injury. And generally, when the GGT level goes to above 3 times the upper limits of normal, and oftentimes when it's sort of in the 100 or higher range, I think most clinicians will escalate the oral steroids at that point in time from 1 milligram per kilogram to 2 milligrams per kilogram. And then if we see further significant elevations in AST, ALT, as well as GGT, or if we see any movement at all in some of the synthetic function in terms of some of the blood coagulation factors or the bilirubin, I think that's when we intervene aggressively with IV steroids, IV methylprednisolone, and I think many clinicians are now doing repeated dosing over several days, 3 days, to treat that acute liver injury. And I think in the vast majority of cases, it's been very responsive. I think what's been very unusual about these 2 cases of liver failure is that they have had really marked elevations in liver enzymes and increases in their bilirubin values, really dramatic increases in their bilirubin values and their INR values that have been really unresponsive to the IV steroids. And then there's other treatments that are entertained, such as IVIG or sirolimus and other possibilities. But hope that answers your question.
So Craig, is there a well-defined protocol then? So when you see the GGT go 3 times, you start with 2 mg per kg of prednisone. Then when it goes if it continues to elevate, you intervene with IV prednisolone, or methylprednisolone.
Yeah, IV methylprednisolone. Yeah, that's correct.
Oh,
And
and then
when
do you go to IVIG, or is that a well-defined protocol that is generally is a consensus amount around the prescribing physicians?
I can tell you we meet on a monthly basis with over 20 physicians that are experts in AAV gene therapy in Duchenne, and we review the data and the adverse event occurrences, and we're sort of available on an on-call basis to treating physicians. That's through the Sarepta Exchange program that has been set up. And I can tell you there hasn't been a consensus in terms of when to implement with IVIG or sirolimus. We've been having those discussions in recent months, but I think the consensus has been when there's a significant elevation in liver enzymes and GGT, or certainly any bump in the INR or elevations if the bilirubin is beginning to creep up. That's when we implement IV methylprednisolone at a dose of 30 milligrams per kilogram, and I think all the physicians agree with that dosing. I think each individual site may have their own protocol, because there may be other physicians who are on call, taking care of these patients. And so we have to have our protocols set up for our individual sites so that everybody is taking an aggressive and consistent approach to patient care.
Well, thank you for that, Craig. And so another question that I have, and we could go to another slide, but while we're here, so I know Doug and James have talked about adding sirolimus to the non-ambulatory group. So there's several questions here. Why wouldn't you add it to everyone as a precaution? And 2, if the FDA, do you expect that the FDA will agree to that, or will they say that, will they encourage or make it important, or say that you have to do trials using sirolimus now so that we understand this much better?
I can answer
I'm
that
just curious.
question real briefly. And that is, we don't yet know the FDA's willingness to agree to a protocol and implement. We don't yet know. We need to have that discussion with the FDA. We have some conviction that it will be a constructive discussion, because interestingly enough, very recently, the FDA actually asked us if we had considered the use of additional immunosuppression, including sirolimus. So you would think that this would be a good discussion, and we'll have that discussion with them, and hopefully we'll have it very rapidly. One of the arguments one would have is if you required someone to do a study, you'd be dosing the same patients without getting to dose the broader community, and you'd be doing harm to the entire community, as opposed to essentially getting this in place and then doing a registry and getting data from the registry as rapidly as possible while giving access to patients in the US. Anyway, then
Yeah.
the scientific questions I'll leave to others.
Yeah, well, I think in other studies across even neuromuscular disease or other studies that are not neuromuscular disease using AAV gene therapy, there have been pretty well-defined protocols using sirolimus and other medicines. So I just wondered, what are we learning from them? Are we gathering data from across this use of AAV serotypes so that we better understand who we can treat and how we can treat them and keep them safe?
I mean, I can answer that, Pat, and anyone else can take it on as well. But absolutely, I think that the experience of other investigators, both in DMD, but also, as you say, in other AAV programs outside of DMD, has to be very informative of this process. I and others have already spoken to several of those investigators in other programs in the last week to understand their experience. Some of it is published. So, for example, the study of gene therapy in giant axonal neuropathy used sirolimus that has a published protocol. So we are taking all this information on board. We're convening an expert panel and will include some of these investigators who have experience in using sirolimus and other immunosuppression regimes in AAV to, I think, answer some of those important questions, like which population within the DMD population will most likely benefit from immunosuppression, and understand, are there additional risks of immunosuppression that we need to be thoughtful about when we select the appropriate patients for that, and how we monitor those patients. So I think that's a really important step, is to hold that next meeting and get that expert consensus and draw on all of these other programs that you mentioned as well, and then take that information on to the FDA to have a further discussion.
Have you scheduled the expert panel?
We are polling at the moment, Pat. We are very hopeful in the next week or two we'll have a date nailed down, and believe me, that's moving very quickly to get academics' calendars in line. And I think it's a testament to how everybody, both within Sarepta but also outside of Sarepta, is committed to trying to find answers to these questions.
And just so we're clear, aspirationally, our goal would be to not to have the date nailed down in the next 2 weeks, but to have the expert panel in the next couple of weeks. Now, that may be overly aspirational, and you've got to get the right people, not just prioritize getting the meeting held. But we're trying to do this as fast as reasonably possible with the right experts in the room, so we get genuine insight.
Yeah, no, absolutely, Doug. So that's what I meant to say was that it would be the next 2 weeks we're aiming to hold this meeting. The polling is out there as we speak.
So the two young men that we lost, I can't imagine how their families feel, having lost my own sons, but having no opportunities for a therapeutic or a medicine. And now we have these opportunities, and still losing is just heartbreaking. So I wonder if you could share with us, and you're allowed to say no, were these young men similar in their genetics? Were they similar in their the elevation, or the elevation over time of those liver enzymes and those liver values? What were, if you're able to share, the similarities? Have you compared them? You mentioned in that first young man that died, CMV, the CMV virus. And I guess in all of that, are you doing more testing to evaluate these children or these young men as candidates for the therapy, so that you have a much better picture of their B cells, their T cells, whether or not they've been exposed to an infection? Are we doing more diligence before we consider including them in this therapy? Greg? Anyone?
Yeah, no, I can. I can certainly speak. I'm mindful of privacy considerations with these two patients, but I can sort of speak to similarities and differences, I guess, without specific details. I think fair to say that the cases were very similar in terms of patient age, ambulatory status, obviously, as we've discussed. The clinical courses were very similar. The initial acute liver injury was seen in both patients within that sort of 4 to 8 week window that we have seen consistently across our studies and commercial use. And then the subsequent development from the acute liver injury to acute liver failure was also very similar in time course. Sadly, both patients passed away in a very similar time period as well, really within 3 months of treatment.
Thank you. The first boy that died, I think some months back, was in the commercial therapy. So I'm just wondering about the real world data that you're collecting and reporting to the FAERS database, the FDA data collection, where there are these reports or these instances that are reported. Are all of your physicians required that are delivering this therapy required to report anything that they see to that database, or how does that work? Or is it, if they think about it, is there some knowledge to do exactly that, so we know whose liver enzymes or what side effects we're seeing in the commercial arena, so that we can track sort of the breadth and scope of what is happening in the commercial arena?
I mean, so I can speak to our safety approach, which is regulatorily mandated. You know, it's a legal requirement of ours. We would do it anyway, but it is not something one can choose to do or not to do. So we have a requirement to seek reporting of adverse events from our medicines, to follow up when these events are reported to us under very strict timelines, depending on the severity of the event, with the treating physician to get as much detail as possible. We also, again, will report all of the adverse events to the FDA, and then how quickly we do that depends on the severity of the adverse event. But in the case of fatality, that's a matter of days. We will, if we learn of the event, we have to report that in. We'd always report that into the FDA within 7 to 15 days, depending on whether the event occurs in a trial or the commercial setting. So we maintain all of those safety reports in our own internal safety database, and part of our process is also to cross-reference that database with the FAERS database. So if we see a safety case on FAERS that we don't have internally, we'll add that to our own safety database, and our team will follow up on that to make sure we have all the relevant details. Similarly, if we see a case on our safety database that isn't on FAERS, then we will report that into the FAERS system. And obviously, we're continuing always in an ongoing fashion looking at new safety signals within our own safety database and anything we see externally, to see whether we need to take additional measures in terms of patient eligibility for treatment, or in this case, whether additional risk mitigation like immunosuppression may help.
Thank you. So, and for these individuals, was it a very similar time from infusion to the change or the elevation of the liver enzymes, the liver values, and then their ultimate death? Was it the timing the same in those two instances? Exactly the same?
Well, so there were, I think the first patient, I think, had enzyme elevations in week 4. The second patient in week 7. They both passed away within a few weeks after that. So it's all in terms of, as I said, you know, in terms of the acute liver injury, we see that very consistently between weeks 4 and 8. Both of these boys were in that window for acute liver injury. These are only 2 examples of acute liver failure, but they both behave similarly in the sort of progression to liver disease over liver failure, sorry, in a period of a small number of weeks before passing away.
Thank you. And have you heard from the FDA, or do you have a timeline when you might be hearing more from the FDA or a response to your questions?
So the FDA have asked us some further clarifications. We had an opportunity to clarify a process with them as well. So they're keen for us to do the expert committee first and then follow up with a meeting with them.
So and does the FDA then have access also to your commercial product and who's been treated and whatever SAEs or AEs that or liver enzyme changes over time? Do they look at that data as well?
Yes. So I'm just trying to answer that question fully. So all of our commercial adverse events that we're aware of, we report into the FDA. So they are aware of all of that data. We also have a periodic reporting requirement where we accumulate all of the data from various sources into a single document that's submitted to the FDA and to other regulatory agencies around the world. And then they'll also keep their own database based on spontaneous reporting into them. And that's where the FAIRS comes into place as well. And as I said before, we do our best to keep our internal database and the FAIRS as consistent as possible by keeping up to date with what's on FAIRS and then also keeping the FDA up to date with what's in our database.
And this is both US patients and ex-US patients as well, reporting into FAIRS?
So FAIRS is US only. Just sort of think this question through. So we will report to the FDA on trial experience globally. The commercial experience outside of the US, I may have to come back to that, certainly would be reported in via our partners at Roche to the local regulatory agencies. If there was a major
if we become aware of an event, we report it to the FDA.
Yeah.
Okay. Ex-US. Okay. All right.
And
We're about time.
Pat, I think one other point in terms of this expert panel that Sarepta is convening is I think there really is going to, I'm sure, be discussion about early treatment with sirolimus or immunosuppression, really prophylactically, even prior to the gene therapy administration. So prior to the time of induction, and then continued treatment, probably for months after the gene therapy. And then I think the other discussion will be the treatment of acute liver injury after, once it's occurred, because obviously there's going to be, there's going to be ambulatory patients that may develop acute liver injury that also may be candidates to be treated with sirolimus. They may not be treated at induction or in a preventive fashion. Some physicians I've talked to actually will be considering treating all of their ambulatory and non-ambulatory patients with sirolimus. I think that's going to be, somewhat variable. It's an FDA approved medication. So treatment centers can develop their own, treatment protocols. Obviously it'll be informed by what Sarepta and the FDA and the group of experts come up with in terms of a treatment protocol. But in terms of decisions about who will be treated with sirolimus, I think will be made at the at the patient level at the treating sites.
Yeah, Craig, it's so terrifying as a parent because we're going to see this on social media, right? We see right now on social media where some children are quarantined for a period of time and their parents are doing all the things necessary to be in a quarantine, in quarantine, right, with your child. Then it varies with some children going back to school, shortly thereafter after an infusion. And so then you as a parent kind of say, well, what's right here? What's the proper thing, right? What's the best thing to do? What am I doing? And in the same way, when we see variability across the clinical community, I'm not saying it's wrong. I'm just saying it's hard as a parent when Craig McDonald says, do this in this order. And, another physician doesn't do it in that order. So then that increases our anxiety about what's the right thing. And I will say to you, as a parent having lost two boys, there's not a day that goes by that I don't think, if only I knew that they were in heart failure, if only I knew that they were there, and we're playing the same role here, right? If only I was with Craig McDonald and he did X, or if only I was with this other physician and he did Y. So I think this is where we really need to have some well-defined guidelines and protocols so that we know as a community, what is, in terms of consensus, the best steps forward, so that we don't feel this, if only I was somewhere else, maybe I would have those minutes back with my son. And that's, as a parent, what I'm going to ask every single day, right? So and I'm begging you in this expert panel to think through what happens on the other end as I learn this one does this, somebody else does this. What should we do as a community? What are the questions we need to ask? And then put the responsibility on us, right? Because I know if somebody came to draw blood and they had a hard time and they couldn't get the blood, I might say, he's fine. So what are the imperatives for us as a community? What do we need to know and do? When do we need to maybe call you every 15 minutes, Greg? And I've done that in the past. So when do we need to harass you as well? This is a community. We're in it together. We want the best for our patients and family members. We want them to stay safe, and we want them to have more time and more function for longer periods of time. So I beg you in this expert panel to get protocols that we can understand and that we can do our part to keep our kids safe and healthy in an environment where we are having therapies that are going to be helpful in terms of preserving function. And with that, I'm going to turn it over to Eric and say, what are the questions I missed, Eric, in this discussion? I appreciate all of you staying on for this long of time, but I think this discussion was really meaningful and helpful. Liz.
Yeah, hi everyone. Thanks, Pat. The only kind of things that I think we maybe would be helpful for clarity is I did see a number of questions in the chat coming through specifically around the ambulatory status and questions of, is this, acute liver failure tied to something unique about being non-ambulatory? Or is this more of a looking at non-ambulatory as more disease progression? And is there a risk for that within the ambulatory population, even if it hasn't been seen yet? So I think a little bit of clarity there would be helpful.
So it's the latter, Eric. So we have seen acute liver failure only in these two cases in older non-ambulatory boys. That's led to the decision to suspend shipment on those cases. We are currently hypothesizing that there is an association between disease progression and a risk factor for moving from acute liver injury to acute liver failure. Things like chronic steroid use, for example, may be a factor. These are things that we're still looking into and we'll discuss with the expert committees that we're pulling together. It still remains a potential risk in the ambulatory population, I think, based on the fact that we do see acute liver injury with the same frequency in that group as we do with the older patients, even though we have not seen acute liver failure despite pretty substantial experience there.
Thanks. And then I think the other thing that we kind of saw coming through and a little bit reflective of, we've obviously done patient preference work with the community. And so we understand that there is, typically a high risk tolerance in the community for therapies that may have some risk in exchange for slowing disease progression. But of course, want families to be as informed as possible when making those decisions. So there were some questions specifically about the details of those timelines for those two individuals, when kind of more specific details would be made available. And I think families were asking in terms of some of the ones who may be kind of in that more recently treated window, who may have concerns about if they see some of these liver enzymes being elevated. And then for those, ambulatory families who are still considering treatment on ELEVIDYS, just as they kind of make that decision with their clinical team.
I'm sorry, I had a little bit of a glitch there from Wi-Fi again. So this was a question about timeline of liver.
For the two deaths, when specific details of timelines would be made available to the community. And I think with this too, always helpful of, maybe not just at the clinical level, but for families knowing that we have a lot of variation in kind of health literacy in the community.
So Eric, I think James addressed that from the perspective that both cases were a similar course. So within the first couple of months post-dosing and within that 90-day window. So everything happened within that period of time that is really critical for monitoring and understanding what's going on. What was unique about these two tragic cases is that they progressed to acute liver failure, which we had not seen before. So that really is what drove this being a signal and why, as a company, we've reacted so quickly to stop and learn more. But from a timing perspective, does that answer your question that it's within that 90-day window post treatment with ELEVIDYS?
Yeah, I think the question they were maybe looking for, the granularity within that. I'm trying to read from the questions that are posted there. So I have a feeling it's more of that is, yes, they know the 90-day window and that's probably the mark they're looking for, but I think maybe just, I think again, families, just as much information as they have tends to make them feel more comfortable in making the decisions and kind of going through that process.
When we get through the expert panel and we have better view on protocol and the like, we'll share all of that with the community when we're permitted to. We've talked to the FDA as well and the like. So
Craig, if you wouldn't mind reassuring families who've been dosed within the last 30 days or 2 days for that matter, how to reassure them, what to look for. I can imagine that if my son was diagnosed shortly before this occurred, I would be really worried about what am I supposed to look for? What am I supposed to do?
Yeah, well, I think really the critical issue is really diligent laboratory monitoring on a very consistent and regular basis, as per the protocol of your treatment center, which I'm sure most treatment centers have very rigorous monitoring in place. And I think, if there is a slight signal, sometimes we have to increase the frequency of that monitoring so we don't miss a window of opportunity for treating really in a expeditious manner. I think obviously there's patients that are non-ambulatory that have recently been dosed with ELEVIDYS, and I think you need to have discussions with your physicians as to whether or not some consideration of sirolimus at this point in time may be something at least to discuss. I don't think Sarepta is in a position to make a recommendation. They're convening the expert panel, but I think realistically there are patients that have recently been infused with ELEVIDYS that are doing fine now, that are within that first 4-week window, that we need to make decisions on as to whether we want to add immunosuppression even after that initial induction period of time.
Back to you, Eric.
I think the only other kind of a big piece question that I saw was around, so we talked a little bit about, or James, you talked a little bit about the ENVISION study. So just for kind of community expectation of, when is the anticipated completion of that study and that families could potentially see data from that? And I know you talked a little bit about the blinded data. Do also, I think, maybe want to address some of the challenges of sharing blinded data in terms of the interpretability, because I'm sure that's also a question that families will have of why can or can't we do that?
Yeah, sure. I mean, from a blinding perspective, it's blinding is the gold standard for ensuring that the trial results properly represent treatment effect versus anything else, any other bias that might be in the study. And for that reason, regulators take it extremely seriously. And so to unbreak the blind and look at the data, understanding which patients got treatment and which patients got placebo would likely render that trial of limited to no value to regulators. And fundamentally, that's going to be a real challenge for then making this treatment available in the long term, both in the US and outside of the US, to non-ambulant patients. So that's why we are prioritizing conserving that blind, which I appreciate is not an answer that people on this line want to hear, but we are doing everything we can to look at the data within the study whilst preserving that blind. So we both, as I said, I think earlier with Pat, we have patient level real-time monitoring of things like lab results. Clearly, if we see big LFT, liver function abnormalities, we are assuming that's probably an on-treatment patient issue. On an individual patient level as well, like if there becomes a serious problem on a 1 or 2 patient level, we do break the blind. So in this circumstance, for example, with the Envision patient, we did break the blind, which means find out what treatment this one patient was on. And a study can do that for 1, 2, 3 patients and still be acceptable to regulators. We do it for everybody, and it will become unacceptable to
James,
regulators.
I had a quick question for you in terms of the conduct of the Envision study. If you're implementing a sirolimus treatment regimen in the context of a double-blind placebo-controlled trial, how do you do that in terms of maintaining blind? Obviously, you're only going to be treating the patients randomized to active treatment in the part 2 of their trial. You're not going to be treating placebo patients with sirolimus. People are going to have to be monitoring drug levels of sirolimus and so forth. How do you actually logistically do that in the context of a double-blind placebo-controlled trial?
It's a great question, Craig. It's yes, that is our current plan. We've paused dosing globally in the Envision study, which makes answering the question about timeline to getting data difficult at the moment, Eric, because we have to implement a protocol amendment. So we're going to add in sirolimus to non-ambulatory patients in this study. That will have to be approved by regulators everywhere around the world where the trial is being run, and then the trial will get up and running again and recruiting again. The technical question around blinding, Craig, is a great one. We're looking into it at the moment. There are examples from other treatment areas where you can have processes to sham a placebo version of sirolimus and sham the dosing. Maybe getting a little bit technical at the moment, but we're looking into it. There are precedents in other treatment modalities where you can maintain the blind even when you have to titrate the drug in the placebo arm.
Yeah.
So I'll just be clear, we wouldn't intend to add sirolimus to the placebo patients. It would be placebo sirolimus.
Great.
Other questions?
I think we got through, I mean, a lot of them you addressed when you went back through the slides that were being asked in the chat as we went through. I think good to reiterate, though, again, that while we are at conference, there will be time available for if there are additional questions to meet with the Sarepta team and ask those from the community.
All right. So with that, I hope that you'll come back to this community and talk about what the FDA has to say, what the expert panel has to say in terms of the affairs database and some of the other events that you're seeing. If we could understand at what ages these are occurring, are they more frequent in the older boys, older being above the age of, I don't know, 8 or 10, and are they non-existent or much less frequent in a very younger age? We'd love to have another call, another discussion, and I know you're going to be at the conference and answering questions for people who are here. So to that end, I wanted to thank Wendy and Doug and James for joining us at this meeting tonight. Thank you for taking time for this webinar. And Craig, always thank you for your insight. We appreciate all you do for us. And we certainly do like to harass you with 1,000 questions, and we'll continue to do so. So thank you to Sarepta. Thank you to the PPMD team for getting this up and running in the middle of our conference coming up starting on Thursday at noon. So thank you so much for joining us, and we'll come back to you with additional information. To our community, you're the best. Always bring your questions and we'll try our very best to get them answered. Stay safe.
Thank you.
Thank you.